Welcome all to this video lecture. I am Dr. John Milton, working as an assistant professor in the Department of Microbiology, St.Joseph College of Arts and Science, Autonomous Cuddalore. So in this video lecture, we will discuss about grass-issued disease of sugarcane from the subject of soil and agriculture microbiology. First of all, we will see what are the symptoms associated with the grass-issued disease. So initial symptoms appear in the young crop of 3 to 4 months old age. Later, white or yellow tillers appear in large numbers below these leaves. You can see the proof is tillering. So here the leaves are yellow in color and numerous in number. So this disease appears in isolated clumps. You can see these symptoms in the isolated place on a land, on a field. The affected clumps are stunted with the premature proliferation of axillary buds. So they are very stunted. The axillary buds are very close to each other. So this is axillary buds. You can see the axillary buds very close to each other because of the stunted growth. The can formation rarely occurs in the affected clumps. Sometimes the can may be formed with short distances between the axillary buds. Sometimes the can may not be formed in the affected plants. So the leaves become pale yellow to completely chlorotic, thin and narrow. Chlorotic means the loss of chlorophylls. So because of the loss of chlorophylls, they will be yellow in color, thin in nature, narrow in nature. So you can see the leaves, they are narrower here. The plants appear bushy and grass-like due to reduction in the length of the internodes, premature and continuous tillering. So because of the continuous tillering means branches, development of the branches, as well as the internodes premature, the reduction of the length. So because of this reason, the sugarcane plant will be a grass-like bushy plants. So because of this reason, this is called as grassy shoot disease. The pathogen. So what is the pathogenic organism which are causing this disease? Is mycoplasma like organism, that is Phytoplasma sakari. So this disease is also transmitted by mechanical agents, especially the knife which are used for cutting the sugarcane sets before it is sowing to the field, as well as some kind of insect. Some kind of insect also transmit these pathogenic organism from the infected field to other field, healthy field, aphids, blockhopper, as well as the dodder. A dodder is nothing but it is a parasite. It is a parasitic plant. So it is a thread-like plants. It may grow on other kind of plant as a parasites. This is dodder. This also responsible for spreading the diseases to the adjacent field. What are the management strategies? How can we control the disease? First, cultural methods. Simply we can avoid the losses due to the diseases by cultivating the resistant varieties. So Co 86249, Co G93076 and Co C22, these are the resistant varieties. These varieties will not be affected by these phytoplasma. So we can choose these kind of varieties and cultivate these varieties by for escaping from the disease. Then avoid retooning if grassy shoot disease incident is more than 15% of the plant crop. So if you see the grassy shoot disease incident in the area more than 15%, so we should stop the retooning process. Retooning is nothing but the emergence of the plant after harvesting. So this is called as ratooning. So usually once we planted the sugarcane, once we sow the seeds of the sugarcane, we are harvesting almost 3 years without sowing the seeds again and again. So this is ratoon. If the disease symptoms are visible within 2 weeks after planting, such plants must be identified and that should be removed and replaced by healthy plants. So by which we can prevent the spread of the infection to the adjacent plants. So uprooted the infected plants need to be disposed of by burning them. So we should identify, is there any kind of plants with a symptom of grassy shoot that must be uprooted from the field immediately, and that should be disposed by burning them. Then physical approaches. So some of the physical approaches can be used, can be followed to control the grassy shoot disease. Rug out infected plants in the secondary and commercial seed nursery. Rug out means, roughing means the identification of infected plant from the field and removal of the infected plant, that is called as a rough out or rugging. Simply it is called as rugging. So that is the best way. In the preliminary stages we can avoid the spreading of the pathogenic organisms to the other healthy plant. Treat the seeds with aerated steam at 50 degrees Celsius for 1 hour to control primary infections. So we can treat the seeds. Seeds means that is a seed for the, that is a portion of the sugarcane act as a seed, used as a seed. So we can treat the seeds with the steam of, with a temperature of 50 degrees Celsius for 1 hour. So it can control the primary infection, it can kill the mycoplasma. Then treating them with hot air. The hot air can be passed over the seeds with a temperature range of 54 degrees Celsius for 8 hours. Or we can spray the epidicides, that is a pesticide which control the aphids, which are the agents to spread the diseases from infected field to another field. So by using these kind of pesticides we can control the spreading. We can spread twice in a month once we see the symptoms in the field. Then some kind of chemical can be used. So the spray dimethoate at 1 ml in 1 liter of water to control the insect vector, and also apply the pesticide methyl dimeton 2 ml per liter of water for controlling aphids. So these are all some of the chemicals can be applied. So this is regarding the grassy shoot disease of sugarcane. Thank you for watching this video.